to what, well, my name's Cameron. I'm a professor in the Department of Bacteriology. I've been in Wisconsin since 2004. And we, I think, as you all know, work on leaf-cutting ants and symbiosis. And how many of you have seen the big colony we have on display downstairs? Only 4 of you, though. So if you get excited about leaf-cutting ants, you have to check out our big display downstairs on the main floor of the building. So, yeah, and I'm happy to answer any questions you guys have.
If you want to put your recorders, put it there now. You're not going to pick it up from where you're sitting. That's what I'm giving you.
I feel like I'm in a press conference.
You are in a press conference. This is the press.
Sorry.
It's okay. Go ahead, please.
Who asks questions?
I have a question for
Sure.
you. What made you interested in leaf-cutter ants? How did you get to that point?
How did I get interested in leaf-cutting ants? So I did not grow up with having ant farms or anything like that. So that's not my interest. There are people who do get interested in ants from a very young age and become people who work on ants. So my interests are complex connections of organisms and biodiversity, and questions around why is there so much biodiversity, what shapes biodiversity, what maintains biodiversity, and a lot of what influences biodiversity is species interactions, and in particular pathogens or parasites, so organisms that have negative impacts on their host, like disease-causing bacteria, or beneficial associations, so microbes typically, or other organisms that benefit each other. And so I'm interested in these kind of tight, specific interactions between different kingdoms, different groups of organisms. And the leaf-cutting ant system is a textbook example of a beneficial association, a mutualistic association between distinct organisms. And so I did my PhD on leaf-cutting ants, and that's when I first got started working on them. And it was really kind of being fascinated by the success of this beneficial combination of the fungus that they grow and the ants themselves. So I don't know how familiar people are with leaf-cutting ants, but it's not unrealistic to say a mature colony of leaf-cutting ant could fill the volume of this room, with the forest floor being at the ceiling and them being 10 feet deep underground, with hundreds of interconnected garden chambers with 5 million ants. So it really is an example of a mutually beneficial association resulting in real ecological success.
Question? Dakota, what's the next logical question?
Can you tell us a little bit about what you discovered?
Well, so I mean, I don't know how familiar people are with researchers, but I mean, I have 14 full-time people working in my lab, and I've been working on leaf-cutting ants for 15 years. So we've published maybe 60 papers on leaf-cutting ants, maybe 70 papers on leaf-cutting ants. So that's a hard question to answer. I could lecture for 3 days about stuff we've learned about leaf-cutting ants. So is there a particular aspect of leaf-cutting ants and discoveries that you're interested in, or?
Well, what's the significance?
What's the
What are a couple of the
significance?
major discoveries?
Yeah. I'm wondering, just going off of that, the major discoveries, but the major discoveries that are important for the general public to know.
Okay.
So.
Yeah, I brought a little show and tell.
Okay.
So I don't want to make this a political discussion, but a colleague of mine gave me this yesterday, and it's Obama spent $1 trillion for job stimulus. What jobs did it create? And the number one criticism is $1.9 million for exotic ants.
Oh my God, you made the headline.
Well, it could have been my work, but it actually, fortunately, was not. But I mean, that's a great question. I think it's an important question, and I think my philosophy on science is that kind of pure science and pure knowledge about the way the natural world works is critical, and a critical component of being a advanced society is wanting to understand the world around us. And so I really value human endeavor of trying to understand the world. And so we in our work, ultimately our major interest is understanding how biodiversity is shaped and is stabilized, and so there's a lot of pure science driving components of our work. If you are concerned about biodiversity loss, stability of ecosystems, you need to understand how they operate to manage them, and so one, so there's a pure science perspective that we take, and I think it's safe to argue that almost everything that we depend on is through some form of ecosystem services in the natural world. Our agricultural crops are ecosystems that depend on understanding of biological organisms and species interaction and influence of biodiversity and stability. Water, ecosystem service, food, fiber, wood, all what we depend on is ecosystem services. The other major benefit of what I would argue is that a lot of pure science that we undertake today results in the applied outcomes of the future. And so a lot of the argument I would make against this kind of criticism is that I think our work is a great example of applied outcomes from pure science effort. So I started working on leaf-cutting ants because I was fascinated by complex species interactions. And one of my major discoveries as a PhD student is that the success of ants growing fungus for food, so they farm fungus for food, the success of that is partly through associations with bacteria that produce antibiotics. And so arguably my most significant discovery is that of this association, that the ants not only culture fungus for food, which was known for over 100 years, but that the success of that association is through associating with another organism, this organism being a bacterium that produces antibiotics. So probably none of you are, well, very few of you are aware, but most of the antibiotics we use pharmaceutically are derived from bacteria, and so probably everyone in this room has a relative, or may not be here because a grandparent or a parent would have maybe died from infection if it wasn't for antibiotics, or would have lost a relative through antibiotics. So antibiotics is, had a major benefit to humans. Well, those antibiotics are derived largely from bacteria, and these bacteria produce them in natural systems to mediate their interactions with other microbes or in the systems, in the niches they occur in, and we capitalize on those. We take them from nature and we discover new antibiotics from them. So the ants, like humans, benefit from associating with these types of bacteria for obtaining antibiotics that help protect their fungus garden.
Okay, so let me get this straight. They use the bacteria to help develop this fungus?
No,
Or-
so they're farming, right? And in human farming, we have major problems with diseases attacking our crops, and if we have a major plague of a pathogen going through and killing a crop, then that has major impact on humans. And the Irish potato famine is a. Does anyone have Irish blood in them? So 3 or 4 or 5 of you in this room wouldn't be here if it wasn't for the Irish potato famine. Likely wouldn't be here in terms of the migration of Irish people to the New World. So that's an example of an agricultural pathogen impacting the human farmer. Like human farmers, ants that farm fungus for food, if their fungal crop dies from disease, the ants die. And so they depend on their fungus garden. If their fungus garden dies, they die. And another one of my discoveries is a
To follow up on the ants, though, so do they develop the bacteria within their own bodies, or?
So they actually maintain the bacteria on the surface of their bodies. And these bacteria secrete, so they don't actually produce the antibiotics. The ants don't produce the antibiotics. They support bacteria that produce the antibiotics and secrete them. And these antibiotics help protect and maintain the health of their fungus garden.
So by consuming the bacteria that's already on them, they
No,
secrete it and then it becomes an antibiotic?
No, so the we take antibiotics orally
Yeah.
to deal with diseases internally. Their fungus garden is external to their bodies, so they crawl around in the fungus garden. The bacteria occurs on their bodies, and then they apply the antibiotic externally. So they don't require any internal
Do they
acquisition.
pass that bacteria down from generation, or how does it get on their bodies? Do they go
So-
find it?
So they actually, the queen, so this is a social insect, and so social insects have queens, and these queens lay the eggs, and then the eggs turn into workers, and the workers do the jobs in the colony. And the workers have the bacteria in their bodies, and the queens have the bacteria in the bodies. Within a nest, we've shown that the bacteria is transmitted from worker to worker. So new workers, new adults obtain the bacteria from older adults that have the bacterium. New colonies obtain the bacterium because queens that are produced from the colony go on what's called a nuptial flight or a mating flight, and they go and establish a new colony, and they carry the bacterium on their body, so they have it to pass on to their workers. They also carry the fungus that they grow for food, and
We
so
don't know originally how the bacteria became part of it.
the association. I have theories on that and of the origin of it, but we know that the ants have been associating with this bacteria for at least 20 million years, and probably since the origin of the mutualism, which we think is 50 million years ago.
Now, see what happened was Dr. Currie was going to give us another example of his work, but Leanna very smartly said, well, I don't fully understand this
Yep.
yet. So before he moved on, you were smart enough to say, we're not going to write all this, but we need to get it or we won't be able to
Yep.
translate it. So, not that
Yep.
easy. So go ahead, Leanna.
And I actually have another question. This is kind of off topic. Is that okay? Yeah.
Good morning.
So what was your best discovery, most important to you? What was
Most
best
valuable
point
to me.
in your studies for you?
So I mean, a lot of running a research lab as a professor is mentoring grad students who do science and make discoveries and follow up on ideas that partly stem from my work, partly stem from ideas that I have or ideas they have, and so there's a less personal connection as a professor because I'm not actually doing the work. The students in the lab are doing the work, and they have more of a personal connection. So my the biggest personal connection I have is to my work that I did as a PhD student, which is very typical of scientists. And I made two major discoveries in leaf-cutting ants as a graduate student. The first is the discovery of these bacteria that produce the antibiotics in the ant system, which we were talking about. And the second is what I actually first started working on in the system, and that is I was fascinated by the fact that the ants are successful and have been successful for tens of millions of years of growing their own food source, so farming their own food source. And in the literature and in the whole field, it was thought that the ants were so good at agriculture that they maintained their gardens, their farms, free of disease. And so I was fascinated by this because in human agriculture, as I mentioned, diseases have major impact. And so I thought, okay, wow. And there's one other important point, is that it was thought that the ants, like humans, maintain a monoculture of a single crop type in their gardens, and that they've been clonally propagating this crop type. So that means that their crop is genetically homogeneous. And so one of the major issues in human agriculture, and why things like the potato famine, Irish potato famine occurred, is in human agriculture we maintain these genetically homogeneous crops, and if you have a pathogen that evolves to be able to be very virulent on that genetic type, it spreads very rapidly and wipes out the crop. And so ants being able to maintain monocultures of genetically homogeneous crops without problems with disease was very intriguing, because I thought, well, that's surprising, because when we do this, humans can't do that. And so I thought, okay, either A, they have mechanisms in place to be successful at doing this, which relates to the bacterium on the ants that I discovered, or there's diseases in the system that people have not actually identified. And so I really started with the hypothesis that there was a specialized group of specialized pathogens that attack and exploit their crop. And that's in fact why I discovered that there's a fourth organism in the system. So ants grow fungus for food, and there's specialized fungal pathogens that exploit their crops. And so those are my two major discoveries, and lots of work establishing it. First, that there's specialized pathogens, and second, that to help deal with those pathogens, they have bacteria. And so when I started in this system, it was thought to be two-player community, ants and fungus, and then I showed that there were two additional critical components to the system. In some ways, my most recognized discovery is the bacteria on the ants and the production of antibiotics, and that's most people would argue what I'm most widely recognized for. But I think in some ways I have more personal connection to the discovery of the pathogen, because that was what partly prompted the discovery of the bacterium in the system as well. So, but they're closely interconnected, so I can't completely.
Why are the pathogens significant? What's their role?
So the pathogens are significant because if the ants don't control the pathogen, their colony is destroyed. So just like human agriculture, if this pathogen has a major impact on the success of the ants. And the reason it's partly fascinating is because I showed that it was co-evolved and been present from the earliest association, and looked at evolutionary patterns of this association. And part of why it's fascinating is and why it's exciting is that it fits what evolutionary theory, like you come up with a hypothesis and you test it, and one of the components of why it feels very personal is that I had people in the field tell me, oh, you're working on pathogens in the fungus garden, well, your PhD is going to be a failure because everyone knows they maintain their garden free of disease. And so if I would have actually believed the scientific literature, I would have said, oh, well, okay, I'll just accept the dogma and move on. And so I think, I mean, it's a good example of science is a process. Over time we gain new knowledge and we build on that knowledge, but the knowledge of today, you know, is the foundation of the knowledge of tomorrow, and you know, part of the scientific process is calling, you know, questioning dogma and testing it and these kinds of things.
Are they adapting the bacteria to fight those pathogens? That's what I don't get.
So the bacteria, the antibiotics that the bacteria produce actually inhibit the pathogen of the system, and we've actually shown that that these are specialized, and that there's what we call an arms race dynamic, is that there's selection of resistance in the pathogen to be resistant to those antibiotics, and that there's adaptation in the bacteria to overcome that resistance. And that has, coming back to the earlier question and my show and tell, the one of the components of the work that I always try to convey to the general public when I give general public talks, and also talk to reporters, is in the importance of peer research, is that we've discovered from my work, I think now 5 different antibiotics from the ant system. And probably all of you are aware that one of the major concerns in biomedically is the evolution of antibiotic resistance, and the issues we have in terms of discovering and having new antibiotics to fight these diseases that are resistant. One of the things that people aren't necessarily aware of is that most of the pharmaceutical companies have given up working on discovering antibiotics, and there's 2 main reasons for that. One is the main reason is the economic benefit of doing this research is no longer there. It costs too much money to develop a new antibiotic to make money off of this, and so they've really kind of abandoned that. And there's 2 reasons why the economic benefit's not there. One is just the length of time it takes to bring it to market, and the more narrow market, you know, treating tens of thousands of people against a resistant pathogen is not a really good market to deal with, for pharmaceutical companies to target, even though for us as a society, having people dying of resistant pathogens, you know, tens of thousands of people each year is too many, right? The second thing is that it's hard to discover new antibiotics, and globally, every year we discover between 20 and 40 new antibiotics. And the reason, and we used to discover globally 100, 150 a year. And so there's been a dramatic decrease. And one of the reasons there's been a dramatic decrease is because what we call rediscovery of known compounds. And so if you go into nature and you isolate the bacteria that we get most of our antibiotics from soil, which is what is mostly done, you have to look. It used to be if you looked at 50 or 100 strains, you would discover a new antibiotic. And now, when you look at those 50 or 100 strains, you're rediscovering already known compounds. And now it takes, the estimate is between 10,000 and 100,000 strains being screened to discover one new antibiotic. And so it's a huge issue.
So it's different bacteria with the same antibiotic?
So it's different bacteria that are producing the same antibiotic, or it's the same bacterial species that is widely distributed that is easily isolated, and both of those issues. Yeah.
So what's the goal of, we've talked about the symbiotic relationship between ants and the crops. What's the goal of your research to understand how humans can take that process and use that to benefit society beyond antibiotics?
So let me say one other thing about the antibiotics, and then I'll answer your question. So we've discovered 5 new antibiotics from ants. One of them is passed an anti-cancer screen in the NIH. So these compounds that we discover are used as chemotherapy agents, and so there's the benefit of that. One of our compounds is high activity against malaria, a major disease, a major killer of humans. And our five discoveries represents a significant proportion of the new antibiotics that are discovered nationally or internationally. The other thing that is important to know is that our 5 new antibiotics are screening from about 50 strains of bacteria that we've worked on. So our success rate of discovery is 1 new antibiotic per 10 strains versus 1 of 10,000 or 100,000. So the significance, the broader significance, is the identification of a new source for discovering many new antibiotics. And if you have a system where you have an arms race where you have selection for new antibiotics because there's resistance evolving, that results in greater diversity in the antibiotics that are being produced. And so we think the source has great potential for discovery. To answer your question, we don't do anything about trying to inform on human agriculture because we don't have strong interest in that area. But we have another applied outcome that comes from thinking about pure science perspective, and that is related to energy. Everyone's aware that energy is a major issue right now, and there's a lot of investment in bioenergy work. And bioenergy, the major emphasis on bioenergy right now is what we call cellulosic bioenergy, or biofuels. And currently we can produce a biofuel from the starch or sugary parts of plants. So you take corn, you don't take the body of the corn plant, you take the cob of corn and you take the food part off that cob, and we can make ethanol from that. And so there's a lot of controversy around biofuels because it's a competition between food production and fuel production. But the vast majority of the energy stored in that plant is stored in the body of the plant, not in the food part of the plant. And so cellulosic ethanol is about getting the energy from the corn stover, the rest of the plant. And currently, so the process of making a biofuel is it's like fermentation, producing alcohol. We're very good at that. We have lots of ability to that, especially here in Wisconsin. So if you have but the yeast that produce the alcohol can only feed on simple sugars. So food represents simple sugars. We can digest it. Yeast can digest it, and they can then grow on that simple sugar and ferment and produce ethanol. The green part of the plant is composed of complex sugars. So they're not simple sugars, and very few microbes. We can't eat corn, the green part of corn, right? We can only eat the starchy sugar, simple sugars. The green part of the plant is composed of a lot of what we call recalcitrant, difficult to decompose material. And the reason it is because plants over evolutionary time have evolved to protect themselves from being broken down by microbes, or they wouldn't exist. And so to produce a lot of energy and bioenergy, we need to break down the complex sugars of plants into simple sugars that then our yeast can ferment to produce the biofuels. And currently, what we need is enzymes to break these bonds between these complex sugars to make the simple sugars. And currently, our technology to do this is limited, and it's inefficient. So it's economically not viable to make a biofuel from the green part of plant because the enzymes are not very efficient. Well, leaf-cutting ants are called leaf-cutting ants because they cut leaves and they put them in their fungus garden. So the ants don't feed on the plants, they actually feed on the fungus. So the microbes in their garden break down the complex sugars of the plant grow and produce simple sugars for the ants. So we can tap into the ant system for potential enzymes that could then be used to produce biofuels by taking these microbes, getting their enzymes, and contributing to biofuel production.
So it's the fungus that break down the complex sugars?
Yeah. Well, and we've actually shown there's a contribution of bacteria that were previously not known in the fungus garden as well.
And how important is that to breaking down the complex sugars, the bacteria? Is that the main?
So the fungus is the major plague, but bacteria contribute components as well. Yeah.
And that's something that we ought to look at in terms of like making that into an alcohol so we can use that energy.
Yeah. Research on the area of making new enzymes for biofuels is focused largely on a small subset of organisms, and it had not been done previously, and we do it in the lab. Part of the reason it hasn't been done previously is that a lot of people who do this very applied science do not actually know that there's ants out in the world, exotic ants that cut leaves and convert them into energy. And so it's an example of understanding pure science that can have a applied outcome that is very significant. So we actually have submitted a patent application with 16 or 26 different conditions of new enzymes that could be used in biofuels.
28 separate enzymes?
26 different claims.
So are these the only species? There are multiple species of like fungus farming ants. Correct. So are these the only species of ants that can do this, or are you looking at the broad spectrum of
So we think that farming a fungus by ants originated about 50 million years ago. And so that probably occurred in one species of ants with one species of fungus. And so the origin of that was probably, just these two combinations. So then 50 million years later, we can look at and say, okay, what's going on now, and inform us on the evolution. And what we know from studying ants and fungus now is that there's a lot of diversity of ants. There's over 230 species that grow fungus gardens, and that different species of ants have different crops that they grow, different species of fungi, and that more closely related ants have more closely related fungal crops. And so this reflects a pattern that we call coevolution. So as the ants diversify and speciate into new species, the fungus they grow diversifies as well. And so the other thing that's important in this is that the first farming in ants was more, what some people might call more primitive farming. Small colonies, very small crops. And then it evolved over time into this very large scale, massive colonies. But there's a lot of diversity of ants that still have what would be viewed as more of the primitive form of agriculture. And I'm doing primitive because as an evolutionary biologist, you don't call something primitive because that's like a human perspective, right? And ants that grow big colonies aren't any more advanced than ants that grow small. They've actually shared the same amount of evolutionary time. They just have more evolutionary changes. So differences across this association, there's major differences, including colony size. But there's also major differences in the types of substrates the ants use to grow their fungus garden. And so we can, by studying different groups of ants, we could get different enzymes and also find different, contribute in different ways to processing biofuels. So we study much of the diversity of the fungus growing ants. Not all 230 species, but we can study different parts of the diversity. And so we can study species that represent more kind of what we call the ancestral or more primitive form, and species that have the more advanced form.
So does that apply to antibiotics as well?
Yeah.
Biofuel?
Yeah, so we know that the different groups of ants have different, as I said, different crops. They also have different pathogens that attack their garden, and they have different bacteria that associate with them. And so by looking across the evolutionary diversity, the biodiversity of the group, we can discover new antibiotics as well. Yeah. Great question.
We have what we need on the ethanol, on the fuel question. We kind of stopped there. Do we have more questions about that before we?
The only other question
Well, James, let's, okay, go ahead.
well, I was gonna say the energy that the ants use to produce this fungus, what's the energy outtake to intake ratio? Has that been looked at?
So it's leaf input, right? In a mature nest the size of this room can defoliate a mature eucalyptus tree overnight. So there is kilograms of dry weight of plant material going into the nest every day. Massive amounts of leaf material. But there's also an output, and the ants take out from the bottom of their garden old garden material, and they place it in these refuse heaps that are very large. And there's actually tens of thousands of ants that their job is to take out the trash, take out this material. And that material we've shown is only partially degraded plant material. So the plant material going in is not completely decomposed by the fungus garden. It's only partially decomposed. And we think energetically it's more efficient for the colony to bring in new leaf material than to completely decompose the most recalcitrant, difficult to decompose components. Energetically, we know that there is very rapid growth of the colony in very short periods of time, so they accumulate garden biomass and new workers very quickly, and so there's a massive input of energy into the system.
Why do you laugh? So to what? There's some logical questions here on the ethanol plant that I think they're still hanging.
So, is it in the future that we're looking for these enzymes that are going to break down cellulosic matter, and do you somehow see like potentially what could this turn into? Like, could it turn into something like ethanol, where it becomes, developed into ethanol plants? And
So the Department of Energy has funded Wisconsin in combination with Michigan State University what's called a Bioenergy Research Center. It's called the Great Lakes Bioenergy Research Center. I don't know if people have heard of this. Have people heard of this? It's a $125 million research grant that was just renewed for another $125 million. And this money is being put in by the DOE because all focused on cellulosic biofuels, because energy independence, contribution towards energy independence is tapping into renewable sources, and cellulosic biofuels is the major source, and so there's British Petroleum has invested $500 million in biofuel, cellulosic biofuel. When big oil companies are putting money into research in this area, you know that there's an importance and a market there. Our patents are on particular enzymes from our work that where the enzyme could be integrated into the process. And so currently there's plants that are test plants that are working on taking cellulosic biofuels and applying different enzymes to get simple sugars and fermenting it. And so our goal is to, hopefully contribute enzymes that could be integrated into that process. And so then if our enzymes are better than the current enzymes and are economically valuable, then companies will build on our patent, take our microbe, or take the gene that produces the enzyme from our microbes and mass produce the enzymes on massive scales.
And if this happens, you're able to process cellulose, which means that you can process anything with cellulosic material,
Yeah.
any plant basically,
Yeah.
that people cannot
Yeah.
digest.
Well, so the major goal is for biofuels, right?
Right.
Because it's not, but there is a human food connection to it, and that is actually using the enzymes as an additive for cattle production, for as a feed supplement to increase the efficiency of cows converting cellulosic material into energy. So I've never really thought about whether this has implications for feeding people, but I mean, if you can break down complex sugars from plants into simple sugars, you can use those simple sugars in diverse ways. So I mean, someone could develop a method to convert that into a food source, but
Because I only say that because ethanol is so, on the rocks, because it's also a food source and people we're using food for fuel.
yeah. So the part of the Great Lakes Bioenergy Research Center has what's called a sustainable component to it, and their research is to ensure the whole process of producing the plant and converting it into fuels is environmentally sustainable. And also, they take economic models into account in terms of support for farmers and these kinds of things. But if cellulosic ethanol production was efficient, then all the starchy material that is food, like the leftover corn material, would not be required and could be used for fuel. So there's not direct competition, right? Because it will be used on the non-edible parts of plants. But having said that, the reason sustainable components of research into the sustainable components of this is important is that energy right now is partly passed back into in nutrients passed back into the soil, right? And so if you're continually stripping all of the material off the field, that has issues in terms of sustainable agriculture.
Yeah.
So that has to be taken into account. But if you have a viable process, it becomes there's more options of how to deal with this.
What is the potential of these enzymes to really make a difference? I mean, do we have any sense of the significance of these enzymes
From our patent?
To find a way to break down cellulosic material?
So part of the reason this is inefficient currently is because it's a complicated process, and there's a bunch of different combinations of enzymes that are used. And so our patent, the reason we have 26 different claims is that we claim individual enzymes and we claim combinations of our enzymes. And it could be that one of our enzymes, some of our enzymes have very high, what we call specific activity, so they're very efficient and much more efficient. They appear to be more efficient than the current standard. And so, I'm somewhat optimistic that our enzymes have potential to have impact. But it is a complicated process. But much like our antibiotic work, we've taken our discoveries in this area and said, okay, well now we know, we didn't discover 5 antibiotics from the ant system to begin with. We discovered 1, and then we used that knowledge to go into other systems and to make new discoveries and discover additional ones. We're taking what we've learned in the ant system and looking at other systems and finding even more efficient enzymes. And so I'm optimistic that our approach that's really driven by evolutionary biology theory and is very novel has the potential to make some real valuable contributions. So,
how frequently are these enzymes being discovered, just on a grand scale?
On a grand scale, so a small proportion of microbes will grow on this plant material, and so there's not a lot of diversity that will grow on it. A lot of these microbes, all these microbes that grow on it, are producing these enzymes. So it's easy to discover these types of enzymes. It's hard to discover enzymes that meet the current standards. So that's very rare. So our patent application is actually centered around one particular bacterium that we have high interest in, and that bacterium, in our review of our paper that we submitted after our patent went in, the reviewers recognized that this bacterium was of high promise and was one of maybe a handful of bacteria globally that would be a high focus. So we've actually discovered 4 or 5 related bacteria that are even better than the one that this patent has actually been submitted on. And these microbes are all from insects that obtain energy from plant biomass systems like the ant system.
Bacteria then produces the enzyme that breaks down the sugar.
Yeah.
Just to make sure we all, okay.
So they, I mean, it's a cell, right? And they secrete the enzyme into the environment, onto this plant substrate. The enzyme cleaves away at the complex sugar, making simple sugars. And then the bacterium or the fungus or both then bring the simple sugar into their cell to consume.
Are the more beneficial bacterium that you're discovering from the leafcutter ants or from other species that you kind of stemmed off to?
So in fact, we have some from the leaf-cutting ant system we're really excited about, but most of the bacteria are from other systems that we've taken, or what we've learned about the ant system and gone and explored.
So entirely different species?
These are not even ants. These
Okay.
are other insects that use plant biomass.
So are these enzymes newly discovered, or are the claims to as what the enzymes do newly discovered?
So the enzymes are newly discovered.
Okay.
Yeah. But some of, like, it gets back to a little bit of the pure sciences. Like, we have actually a honeybee colony right here on the ledge if you, after class, want to look. We maintain one in the lab too. We've gone from the ant system and said, well, ants have problems with disease, honeybees have problems with disease, honeybees would benefit from associating with bacteria that produce antibiotics, and we've gone into the honeybee system and discovered, in fact, bacteria associated with honeybee colonies that produce a novel antibiotic that actually treat or inhibit one of the major diseases of honeybees. And so we haven't submitted the patent, but that compound has the potential to be used for mediating honeybee health. And so that's an example of taking additional steps and building on pure science to have very applied
We
outcomes.
discovered in South America an antibiotic that can help cure the diseases that are wiping out the honeybee colonies.
Well, no, so the discovery of ants in South America led to the idea that we could get these bacteria from honeybees colonies themselves in Wisconsin, and that led to the discovery from the honeybee colonies.
To fight their own disease.
Yeah.
Got it.
And the reason that's important is that if the bacteria is associated with colonies in a healthy way, that bacterium is easier to use to treat their disease because the bees are not going to be sensitive to it, and we can actually promote the growth of that bacterium in the colony and these kinds of things.
So if we're journalists, this story, the general story we've heard, we've seen in other clips. We have not heard this one. So we've got these diseases that are wiping out our honeybee colonies. The farmers are free. Because without honeybees, we don't have pollination. Without pollination, we don't eat. This is quite significant, and I have never heard this. Has it been published?
No.
So that would be news. Now, we're here to get the broader story, but that's kind of how this works in an interview. Suddenly, Dr. Currie just laid something on us we'd never heard before, and that, my friends, is a story. You see why that's news? Because it's new.
And everyone loves honeybees and honey, right?
And we kind of need.
Yeah.
We don't
Well,
need the honey. We need the soybeans and the.
they're critical.
Yeah. So you just asked that question again. So you've discovered with some healthy honeybee colonies that they've got a more active antibiotic, and you've isolated what that antibiotic is and used it then to treat diseased honeybees. It's not.
So the process of discovering new antibiotics, you isolate a bacterium on a plate, but it doesn't like secrete. It secretes out compounds into the media, but you can't look and say, okay, well that's this type of antibiotic. So it takes a natural product chemist, a bunch of time to actually look at that, what's secreted, and see if there's a novel compound. And so we've worked with a natural product chemist at Harvard Medical School who's looked at our strains from honeybees in collaboration with us and discovered that there's a novel compound that's produced by our strain that's not known to science. So it's and then we've taken that pure compound and we've tested it against the major bacterial disease of honeybees and shown it's very highly active against that disease.
So is this specific to the honeybees here, or is this assume that all honeybees produce this in all colonies?
Yeah, well, that's a great question. We don't know the answer to that.
Where
Okay.
did you find the strain then?
In the colony, inside the colony. So the assumption is that, so I don't know if people have heard of the human microbiome, but are people aware of this? So I don't know.
Repeat that.
The human microbiome. So recently, it's become recognized that us as humans are more of a community than an individual, and there's 10 times more bacterial cells in our bodies than human cells. There's 10 times more genetic content in our bodies than of bacterial origin than human origin. If you look at the chemicals in our urine, it's all microbial. A large part of it is microbial in nature. So this indicates that our normal function of our bodies is mediated in a large way by microbes. Every part of our skin has bacteria on it that are specialized. Our mouths, our GI tract, all driven by microbes. And it's used to be viewed as bacteria and humans were bad. Disease, you die, bad bacteria is bad. But the vast majority of bacteria in our bodies, overwhelming majority, is actually beneficial and helps us fight disease. And so if we didn't have the bacteria on our skin would probably be more prone to infection. And this is all new work that's been done in the last 10 to 15 years, kind of a whole new emerging field. Cancers have been shown to be associated with disruption of our normal microbiota. So, some people get a little grossed out, like, your sweetie is 90% bacteria. It's not as, it's but us microbiologists, we think that's cool. Our thought in the honeybee, the reason I'm saying is our thought in the honeybee is that, like humans, honeybees probably have a normal flora of bacteria that provide healthy conditions. And diseased honeybee colonies may be diseased because their normal flora, healthy flora, is disrupted. And so, in fact, the implications of that are when farmers go out to spray honeybee colonies with a general bactericide or microbe, generalized microbe antibiotic, they may actually be doing more harm than good because they may be disrupting the natural normal flora of the honeybees. But if we use a targeted antibiotic that's adapted to the system, then that is much more effective.
So it's basically like a probiotic that you're trying to
It's
get
a little
to
bit
that
like a
other end.
probiotic, yeah, exactly. Yeah. So we actually are working, building on science, in the process of science. We're working with researchers at the pediatrics department looking at the role of normal, healthy microbiota in the throats in affecting strep infections in kids. And so our hypothesis is that disruption of that community is affecting strep infection in kids. So the connection between all the projects we've talked about is kind of the role and the interaction of microbes with hosts.
So is this like the next step, or that's like the next step that your lab is taking?
Yeah. So it is in the broader conceptual framework of systems, but we're doing a lot of stuff in the very conceptual framework of understanding the stability and function of a symbiotic association, like in the ant system, that could then inform on our general understanding of symbiosis that could have implications for human microbiome, human health and this kind of stuff. But yeah, part of it is saying, okay, well, this is what we've learned in the ant system and the bee system, and contributing in that way.
Well, use strep as an example so we can get our minds around this. So from what you've learned, the leaf cutting is, the protocols you developed, you know now what to look for. How did what you learned there then, how does that apply, as Katie was asking, to looking at the relationship of stress and antibiotics and disruption of them?
So it's a little bit of, so there's a couple things, like, a lot of human health research by microbiologists is done by people who are trained to view bacteria as disease agents. So a lot of the field is shaped by people who have a very different perspective than I do. So some of delving into that system is not so much, okay, I've learned the specific technique in the ant system. It's more of I have a different perspective than most medical researchers. And this collaboration didn't start by my initiation. The people in the pediatrics department approached me and they said, well, we know you're doing a lot of what's called metagenomics, looking at microbial communities associated with hosts. We're interested in this question. And so there's some of the skill sets we have that can be applied. But then what happens in that conversation is the perspective of the importance of beneficial microbes in mediating health is what kind of mediates or drives that science.
Called metronomic research?
Metagenomic.
Metagenomic. Could you spell that?
METAGENOMIC.
Just as it sounds.
Yeah. So it's genomics. So Human Genome Project is everyone's probably heard of. Metagenomics is what is done with bacteria, because in humans you have an individual, you can take cells from that individual, you can sequence the genome. Well, bacteria occur in communities of organisms which are combinations of genomes. So metagenomics is the pool or community of genomes that are sequenced and analyzed.
So far, I guess this is kind of like we've all done something different. What has been like the most difficult aspect of your research that you've done?
The most difficult thing, other than getting at the tat.
Well, that's part of it. You have to defend the finding. You have to defend.
No, it's fair. And I think it's important. I think it's important to. I think I take it very seriously. And when I have undergrads do projects in the lab, I tell them, my expectation is we produce a paper from your project, and they sometimes get intimidated. And I'm like, well, we're very fortunate that we can study things that we are passionate about and discover new things about the world, but there's an expectation associated with that, in that we publish our work to build in the scientific knowledge that is the future discoveries, right? And we can't just take the money and play and find out stuff and not produce papers that is what is critical for advancing science. So I take that stuff very seriously. The hardest part of research, I think one of the challenges is, as you go through your career and you become a professor and you advance along, is that the time is, you know, is a limited resource, right? And there is more and more, as as your career develops, as my career develops, more, things, asking for my time. And, there's lots of things that I enjoy teaching. I enjoy this kind of experience. This is a very positive use of my time. But I spent most of my day today writing an annual report on my National Institute of Health. I got a stimulus grant, so I could have been criticized like this person, but my stimulus grant is to discover new antibiotics, and we're actually testing our antibiotics in the mouse model system, so there's no way they could attack us because we actually have very very applied outcomes of our stimulus money. But I have to write reports, continuous reports, and email and administrative stuff that takes a lot of my time. And so one of the challenges is, my lab, as I said, I have 14 full-time people. So I have 3 technicians. I have 2 postdocs, so these are people with PhDs who are going to be future professors, and I have 9 grad students. So that's a very big lab, and that's a lot of managing the lab. Part of the reason my lab is big is because funding in the US is very competitive, and you continually need to do more to get money, and the expectation, the competition continually increases. And so I write grants, and I have to write grants to fund my research. And I actually don't mind the grant writing process, but you are almost forced to get bigger and do more to do your science, because the reason my lab's so large is because I keep writing grants and I keep getting grants, and I have 5 major grants in my lab. And I didn't mean for my lab to get this big, but the problem is you can't say, okay, well, I'm just going to do this amount of science and I will just do this for the next 15 or 20 years and only have 5 people in your lab, because you continually, by competition, need to do more, and then that prompts and asks for more money, and there's this kind of you get bigger or you stop doing your science kind of component to the way things work.
But it's not just the grants. You keep discovering new ideas, and you need people to follow those trails.
Well, yes, and you are, there's a bit of your own passion and drive that results in you getting bigger, right? And I could say, okay, well, I don't have to do that project. But part of doing projects like doing the work with the people in pediatrics is that there's lots of funding for that kind of work right now. And so I don't have funding. We have a small amount of money to do this, but my grant 2 years from now that funds a lot of my work might stem from that research. So you have to have new lines of research going for funding. And so it's a little bit of keeping things rolling and some of the challenges associated with that.
Go ahead.
No, this is not related. Go ahead.
So, what keeps you then driven every day to just keep on pursuing?
To go, what keeps me coming in
What
to
keeps
write
you passionate?
reports and send email.
I guess what just keeps you passionate about this topic?
So, I mean, ultimately, what I'm most excited about is novel discovery. Like, I think there's something I find exciting about discovering something that no one else knows, and obviously, it's more exciting to discover something else no one else knows that is kind of broadly applicable and has application. So that motivates me. I really enjoy mentoring people in my lab. I enjoy teaching. I enjoy communicating about science.
What I wanted So you originally you started out, studying connection of ants and such, but what kind of hooked you to turn and start looking at this fungus more? What was kind of the turning point in your
What
research?
triggered it?
Yeah, what triggered you
So-
to kind of switch, right?
In what do you mean, in terms of the pathogen or the bacteria on the ants, or?
Didn't you originally, you started like mostly, you weren't studying originally this fungus, were you?
So, for the bioenergy, you mean, or no?
No,
Let's
I mean
switch from
originally,
one to
like where
bacteria,
you
something
started.
like- Let's check the internet. Let's look at.
So in science, so we started with what got me into leaf cutting ants. I said I didn't grow up being interested in ants. So some people in biology say, I love leaf cutting ants. I want to work on something about leaf cutting ants, and that's just their passion, and they'll just look at stuff in leaf cutting ants. As I said, I'm fascinated by what shapes biodiversity, biocomplexity, and complex interconnections between organisms, and the role of those complex interactions in shaping diversity, and particularly in beneficial interactions and pathogenic ones. And so, you have an interest in a question area, you need a system to study it. You can't, you need to have biological organisms to ask your question. And so what brought me in the ant system is that it was a fascinating example of a beneficial association between ants and fungus, right? And so I said, this will be an interesting mutualism to work in. And what really made me decide to work on the ants is that I was looking and thinking about projects in the ants, and that's when I read, the ants are so good at agriculture, they maintain their gardens free of disease. So I started with a hypothesis that there was disease in the fungus garden, and that's resulted in the discovery of the fungus that attacks the fungus that the ants culture. And then when I was working on the system, I noticed the ants have a whitish coating all over their bodies, and I discovered they were bacteria, and then I figured out, they're bacteria that belong to a group that are well known for producing antibiotics, and that led to the hypothesis, maybe they're using bacteria to protect their fungus garden. So a lot of the steps, initial step is a specific hypothesis and studying the system. A lot of the additional steps are based on observations or new ways of thinking about the system that result in new questions. The bioenergy work came from a very funny exchange, very funny circumstances. My colleague wrote the proposal to get the $125 million center. And he sent an email to everyone in my department saying, do you want to be involved in this proposal for $125 million? And I said, wow, $125 million. Too bad I don't do anything about bioenergy, delete. Because I work on ants and symbiosis and fungus and bacteria that produce antibiotics, right? And went about my day and said, it's too bad I don't do anything related to that. And then he came down, he wrote me about a month later, and he said, can I come down and talk to you about this proposal? And I said, sure. And he came down, he sat at my office, and he said, we're working on this proposal, and I was talking to someone, and this person said, we need to go to the tropics and isolate microbes to get new enzymes. And he explained to me that the hurdle and the major bottleneck is not having efficient enzymes that break down plant biomass. And he said to me, he said to himself, wow, I have a new colleague in my department who does this weird ant stuff, but he goes to the tropics. So maybe he'll go to the tropics and maybe he'll collect some microbes while he's in the tropics, working on his ants and bringing them back to give to us to work on. So then I said to him, well, Tim, leaf cutting ants feed on plant biomass and forage on massive quantities of plants and have evolved over tens of millions of years to be incredibly energetically efficient. If you're going to find microbes that are good at breaking down plant biomass, you're going to find them in the ant system. And so then it was just a conversation, and I've got written into the grant. It funds 5 people in my lab over the last 6 years, and we have, you know, 26 claims on a patent and a whole bunch of work going on new systems and this kind of stuff.
Tim Donahue is your colleague.
Yeah. You know Tim? Yeah.
He's the head of the Bioenergy Research Center now.
Yeah. Yeah.
Didn't realize he was
Yeah. So you know, it's one of those things where, you know, there's so much science out there that sometimes the communications and conversations lead to new ideas.
So that basically was what spurred the idea of let's look deeper into this breakdown of cellulose.
Yeah, like a month later,
Mm-hmm.
I was an assistant professor at the time, you know, worried about tenure and all those kinds of things. A month later, I was invited to a meeting where the head of British Petroleum's bioenergy thing was in town to interview the University of Wisconsin for their $500 million grant, and I was there, and the chancellor and the vice chancellor and the provost and the dean of the graduate school and Tim and like 5 other people, and I was like, I don't know anything about bioenergy. They wanted to like do all the stuff around the ants, and they were all yeah. I mean, it's just the way science goes sometimes.
I'm surprised it didn't occur to you. They've been doing this for 20 million years, converting plant
Well,
matter to
so,
sugar.
but it occurred to me as soon as I knew the bottleneck was breakdown of plant biomass. I had no idea that the goal of the whole proposal was enzymes to break down plant cell walls, because you know it's that applied stuff.
Before we leave, we've got about 5 minutes. The last time we did this a couple of years ago, you ended by just trying to educate students on the value of UW as a scientific research place. I don't care if you're Democrats, Republicans, teabaggers, what have you. But you wanted to instill in them as journalists a more mature understanding of what we have here, so that if they write critically about it, it comes from a basic understanding.
Yeah, I mean, I think that's a great question. You know, like I'm originally from Canada, and you know, some people are like, well, why are you at Wisconsin? And you know, I think a lot of people in Wisconsin don't appreciate how good Madison is as an institution. You know, if you look at the population of Wisconsin as a whole, and you rank the caliber of UW-Madison for the population of Wisconsin, there's just no reason UW should be such a good institution. We have 800 biology faculty on campus. I came and was recruited from the University of Kansas. I was a professor there for 3 years before coming here. At Kansas, we had 75 biology faculty, so there's 10 times more biology faculty. The patent revenue from biological research on campus is built a third of this building. That biochemistry building is almost all built by patent revenue. Wisconsin Institute of Discovery, patent revenue from biological research. What's made UW-Madison such a strong institution is partly traditional support from Wisconsin for higher education and investment there, but also a lot of really fortunate, outstanding researchers in the past who've made discoveries that have resulted in patents that Wisconsin just happened to have the foresight to develop the Wisconsin WARF Alumni Research Foundation as an institution that develops patents and brings them along. Someone's getting a phone call. And put that money back into the institute, into the UW. And so, I've had a number of outstanding, top-notch, some Ivy League schools approach me about leaving Madison and going to other places. The collection of outstanding biological research and the component of UW is very attractive for people like me to stay and for people like me to come. If you look at the lack of a major city in Wisconsin, like Illinois has Chicago as a population support. The population of Wisconsin is small relative to the caliber of the institution, and so I think it is really an outstanding institution. And I think the other thing that is important to recognize is that UW gets a very small percentage of its research budget from public money. I bring in and spend every year around $800,000, and the university takes 50% of that. So I bring in $400,000 a year to the university. And so researchers who are doing a lot of research bring in more money than they cost for the institution, or net benefit. And I think the number has been over $1 billion in research money that's come into the institution that really helps function, the institution function. That's not something that is, stable. Researchers like myself who bring in lots of money regularly get offers from other institutions to leave, and once an institution starts to go downhill and investment is not there, faculty start leaving, and then you have your colleagues leaving, and you say, well, it's a sinking ship, I'm going to get off. And so one of the things that I think is important to recognize if you're a Wisconsinite is that having world-class international researchers here is a privilege and a fortunate thing. It's not like something that is a given that will always be there. So I don't know if that.
Thank you. No, I like everybody as journalists to understand context of these discussions and debates so that we have a more mature understanding as journalists. It doesn't mean we're going to educate every reader in the world, and it doesn't mean that people like Dr. Currie are always right in their arguments. And
that's true.
This is a political dynamic, but we just need to be more sure about how we report it. Think about it. Thank you so much. Does anybody else have time for one more question?
Can we get some contact info in case we have any follow-up?
Sure. Email will probably be best. Currie, so Curie at B as in Bob, A as in Apple, C as in cat, T as in Tom, so BACT .wis.edu. And if you go to the UW webpage and you type in ants, you probably will find my webpage.
Anything else?
Is there anything you want to add? Or that we should know that you haven't said?
Well, you should look at the ant colony downstairs, and I think I think I made the point of the importance of peer research. My graduate program really would be something that would be a good fit on this line of potential wasted funding. Of course, I didn't get $101.9 million to do my PhD. It was more of like $50,000 total. But it's that pure science that leads to the applied outcomes that I think is very important to keep in mind. And one of the things that I really hope is, obviously if one of my antibiotics is used to treat cancer, or one of our enzymes are used to break down biofuels, I won't have to write another research grant for the rest of my life. So there's a benefit there. But one of my hopes is if we have a really tangible, high-profile applied outcome, is that then I can make the complete link, not just, we've discovered 4 or 5 antibiotics, and these have promises treating disease, but actually are being used in disease, to make that full link of someone who went to Panama to study, esoteric interactions in a little fungus garden resulted in an anti-cancer treatment. I think that's an important point. So
that was a really good quote. I hope you all heard it. That was a really succinct way of saying, here's why this is so important to who I am and how I want to know. So, thank you very much for your time. I know
You're
how busy
welcome.
you are.
No, it's
Good morning.
Russia.